We've almost done 1 week. We are 10% of the way there. Yes, I like that enthusiasm. Before I go forward to the next topic, I just wanted to spend just a minute or so sort of reminding you about the glyoxylate cycle, and then move on. Is this a little loud? Seems a little loud to me. Is this still loud? Better? Worse? Better. Okay? Good. Anyway, the glyoxylate cycle, I went through kind of briefly last time, but the glyoxylate cycle is a cycle that's not found in animals, so we don't have it. It's a cycle that's found in bacteria and yeast, and it's made possible by virtue of the fact that they have 2 enzymes that we don't have. Isocitrate lyase, if my pointer will come on. Come on, pointer. There it is. Isocitrate lyase and malate synthase. Now, the significance of these 2 enzymes, as I noted last time very quickly, the significance of these 2 enzymes is they bypass reactions of the citric acid cycle that do decarboxylation. So because of these 2 enzymes, the glyoxylate cycle does not generate carbon dioxide. So we put 2 carbons in, and by the time we get down here, we still have 6 carbons. 4 carbons here, 2 carbons here. Those 2 carbons get added to 2 more carbons from acetyl-CoA to make malate. So by the time we've completed the cycle, remember this succinate that's produced here becomes malate, becomes oxaloacetate. By the time we get around 1 turn of the cycle, we have 2 oxaloacetates, not 1 oxaloacetate. Okay? And because of that, every turn of the cycle generates an additional oxaloacetate. Well, what was the input material? The input material was acetyl-CoA. That's the only things that came into this cycle. 1 acetyl-CoA here, 1 acetyl-CoA here. Those 2 acetyl-CoAs ultimately become an oxaloacetate. And because they become an oxaloacetate, every turn of the cycle generates an extra one, and extra oxaloacetate can be used to make glucose. We can't do that because we can't convert acetyl-CoA into oxaloacetate in net amounts. Okay? We can't do it. So therefore, bacteria and yeast can use oxaloacetate to make glucose. If we could do this, we would probably have less of an issue of obesity. Because it would mean that instead of using acetyl-CoA to make fatty acids, we could alternatively use acetyl-CoA to make glucose. And glucose we can burn up pretty readily. Okay? So, there's your diet pill. That's a joke. I don't believe in diet pills. All right. Questions about the glyoxylate cycle? Yes?
It does 2 turns to create...
Every turn generates 2 oxaloacetates. Every single turn generates 2 oxaloacetates. Okay? All right. Well, we turn our attention now from a metabolic pathway to some considerations of structure. All right? So this gives you a little break from metabolism. And we'll come back and we'll talk about the metabolism of these compounds I have on the screen later. But for right now, we need to introduce the topic of structure of these because we haven't talked about the structure of them before. This concerns molecules that we find in membranes most commonly. And the molecules that we find in membranes are most commonly called lipids. Okay? Lipids are found in places besides membranes. Okay? But a very common place to find lipids is in membranes. Lipids will always, underline the always, lipids will always have at least a part of them that is very nonpolar. They will always have at least a part of them that is very nonpolar. Okay? Some of them have only nonpolar things. Others have a part that's nonpolar and another part that is polar. But all lipids will have at least part of them that's nonpolar. Okay? Well, lipids turn out to be really important for our cell membranes. And we talk about cell membranes because we need to understand membranes before we start talking about how membranes function. So we need to understand structure before we understand function. This figure schematically shows us what's called lipid bilayer. You probably learned this in basic biology, but I'll remind you if you didn't get this before. A lipid bilayer gets its name because it has lipids. In this case, they're called a couple of names. All right? The names at the moment aren't important, but the lipids in this membrane have a portion of them that is very nonpolar, which are long chains of carbon, shown in green. And they have another part of them that is very polar, shown in red. It's called a bilayer because there are two layers. Here's one layer, here's another layer. And these layers will arrange themselves in this configuration if you put them in water. They will actually, all by themselves, make this structure. Okay? And the reason that they do it is because these very nonpolar tails don't like water. So they're arranging themselves so they are interacting with each other and not interacting with water, whereas the polar portions of them are interacting with water. We have water all the way out here, we have water out here. Well, the beauty of this very simple arrangement, A, is that, first of all, it can assemble itself, and B, this gives a protection for the cell. It defines the boundary of a cell. Now, because of the unusual nature of the lipid bilayer, it's very difficult to move things across that lipid bilayer. So if I'm a cell and I'm needing glucose moves across this lipid bilayer extraordinarily badly, very poorly. If cells didn't have a different way of getting glucose into them, they would die. Because there's not enough glucose that passes rapidly enough to maintain the energy needs of the cell. Fortunately, as we will see, cells have proteins that have specialized functions that are located in the membrane that specifically bring in or kick out specific molecules, glucose being one of them. Okay? Now, another thing to remember about lipid bilayers is that there are some things that move across the lipid bilayer relatively easily. There's only a few. They're all small molecules, and there are 4 of them that we talk about here. One is H2O. Water actually moves across this membrane reasonably easily, even though the middle part of it is very nonpolar. Water moves across it fairly readily. And we'll see that that actually is a problem for a cell. The second thing that moves across very readily is oxygen. That turns out to be very beneficial because cells need oxygen. Okay? The third thing that moves across very readily is carbon dioxide. And that's very good because cells produce carbon dioxide as a result of metabolism. And carbon dioxide is poisonous to them, so they actually let carbon dioxide go out very readily. So carbon dioxide tends to move outwards from the cell. So there's 3: water, oxygen, carbon dioxide. The 4th is very unfortunate. The unfortunate one that moves readily across the membrane is carbon monoxide. Carbon monoxide can move across the membrane very readily. As we will see later, that can interfere significantly with electron transport and cause problems for cells. If we could stop carbon monoxide from moving across the membrane, it would be much less poisonous than it actually is. Okay. This figure, actually that's not the one I want. This figure reminds us of the structure of fatty acids. When I showed you those tails in the last structure that were green and I said were very nonpolar, they are tails of fatty acids. Fatty acids are molecules that have one part of them that has a carboxyl group, and that carboxyl group is ionized, so therefore it's able to interact with water very readily. It's very polar. The other part of the molecule are just carbons and hydrogens, and it's very nonpolar. Fatty acids can be saturated, as you see on the top, meaning that they have no double bonds, or they can be unsaturated, as you see in the bottom, where they have at least one double bond. This guy is mono-unsaturated. If I had several double bonds out here, it would be polyunsaturated. So you hear about polyunsaturated fats. Fatty acids are part of fats. So if I have a fat that has polyunsaturated fatty acids, it would be a polyunsaturated fat. Now, one of the important considerations of the structure of unsaturated fatty acids is that the double bonds that we see in the unsaturated fatty acids are almost always in the cis configuration. In the cis configuration. You hear about trans fat, and that should suggest to you that trans fat contains fatty acids that have trans double bonds. Those don't arise naturally. They're typically arising as a result of chemical treatment of fat, and we'll talk about that later. In general, people say or think, and whenever I say this on the video, I always hear from a bunch of people around the world saying, that's not exactly true, Ahern. But there are links to consumption of saturated fatty acids and things like atherosclerosis, cardiovascular disease. So people try to reduce the consumption of saturated fatty acids and saturated fats. Okay. Fatty acids can be numbered or named in two ways, and I want you to be aware of those different ways. Okay? There's a designation called omega, and there's a designation called delta. The first one that you see on the screen is called omega. Omega is numbered starting at the methyl end of the fatty acid. If we count in to the place where the double bond is located, so this is only a naming system for unsaturated fatty acids. If we count in from the position of the double bond to the position of the double bond, we would say here's a carbon 1, 2, 3, 4, 5. That would be an omega 5 fatty acid. Okay? So you hear about omega 6s, omega 5, or omega 4 fatty acids and so forth, and all they're doing is they're just counting from the methyl end into the location of the double bond. On the other hand, if we count in from the, okay, there's an omega 3, all right, so 1, 2, 3, and there's a double bond. Okay? If we count in from the other end, we create what are called delta fatty acids. So instead of counting in from the methyl end, if we start with number 1 as the carboxyl end, we do the delta naming nomenclature. The delta naming nomenclature is one that's more commonly used in biochemistry than the omega, although they both are used. All right? So a fatty acid that is a delta 9 means that if you start with the carboxyl and you count in 9, that's where the double bond is, that is a delta 9 unsaturated fatty acid. Now the delta designation is commonly used in biochemistry because by the delta numbering, we can say something about what are called essential fatty acids. Okay? We can say something about essential fatty acids. Whenever you hear the word essential, it means it must be in your diet. It means that you can't make it. Okay? Humans have essential fatty acids that have a delta greater than 9. So, for example, oleic acid is a delta 9 fatty acid. Okay? It's a delta 9. There's oleic acid, oleate, right there. We can make that. We can make a delta 9. But we cannot make linoleate. Okay? Linolate is a delta 9, delta 12, meaning it has a double bond at position 9 and a double bond at position 12. We can't make anything past 9. So essential fatty acids in humans will be those that have double bonds past position 9. Linoleic acid, it's Linoleic. These are the salts here. Okay. Linoleic acid, okay, is an essential fatty acid. Linolenic acid is an essential fatty acid. Have to have them in our diet. Yes?
So would the last three on the list all be omega-6 fatty acids?
Would they all be omega-6 fatty acids? So you have to count from the other end. You would have to draw them out and see that. So if you had 18 carbons and this were at 12, this would be an omega-6. This would be an omega-3.
So it's only to the first one.
What's that?
It's only to the first one. Because it has an omega-6. The linoleic has an omega-6 double bond as well.
It has an omega-6, but we count it to the very first one. That's correct. Yeah. So the position of the first double bond. That's right. I'm sorry if I wasn't clear on that. And this would be actually an omega-6. That's right. Okay.
I guess when a food company or we talk about having an omega-6 fatty acid, would it be either of those? Or?
So an omega-6 means it could be anything that has a double bond 6 away from the end. Okay. Now, arachidonic turns out not to be an essential fatty acid. You just confused us, Ahern. Well, it turns out arachidonic can be made from other fatty acids, and we'll see how that happens. But these that we absolutely have to have are linoleic and linolenic. Those guys, if we don't have them in our diet, we'll have problems. Okay. All right. There's linolenic acid. You can see the 2 double bonds. There's carbon 1, 2, 3. It's an omega-3 fatty acid. Okay. And that's what it looks like. Okay. Now, fatty acids, if I take and I link them to a glycerol backbone. Yes, question?
So to clarify, is the omega and delta only for saturated fatty acids?
Omega and delta are only for unsaturated fatty acids. Question was, are omega and delta for saturated? No, the answer is that they are both for unsaturated fatty acids and only for unsaturated, because they're identifying the position of the double bond. Once it has a double bond, it's no longer a saturated fatty acid. Okay, here's glycerol. Glycerol has 3 carbons. If I take that glycerol and I attach a fatty acid to each of those 3 carbons, I make a fat. That's how I make a fat. If instead of linking fatty acids to all 3 carbons, if I take the bottom carbon and I link it to a phosphate of some sort, okay, I create something I call a glycerophospholipid. And that's what you see on the screen. Glycerophospholipid. You also hear these called phosphoglycerides. Same thing. Phosphoglyceride equals glycerophospholipid. When we look at the things that are found in lipid membranes, these glycerophospholipids are one very important component of lipid bilayers. One very important component. If we look at the structure, we see here's a phosphate. We know phosphates are very polar. This is a polar end of the molecule. And here's the nonpolar end of the molecule sticking out to the left. Polar part, nonpolar part. We therefore have something that fits in a membrane really, really well. Okay? Here's another way of looking at that. It's a little bit more complicated, but it's the same basic structure. Here's the glycerol part shown in black. Here are the 2 fatty acids. You notice they form ester bonds with the carbons that are there. And at the third position, in this case, we see simply a phosphate. If we have this structure as you see on the screen, we create something that is a glycerophospholipid. And in fact, it's the simplest glycerophospholipid. That simple glycerophospholipid is called phosphatidic acid, or in this case, phosphatidate. Phosphatidic acid, as we will see, is a branch point for the synthesis of glycerophospholipids and also for fats. We'll see how this can be converted into fat later. Okay. Now, if I take that phosphate that's on the phosphatidic acid and I attach different things to that phosphate, I create new glycerophospholipids. So let's say, for example, I take and I attach serine to that phosphate on phosphatidic acid. If I do, I create something called phosphatidyl-serine. I'll spell that for you. P-H-O-S-P-H-A-T-I-D-Y-L. Phosphatidyl-serine has the following structure. The structure that it has is this guy right here with a serine attached out here on the right. That would be phosphatidyl-serine. If instead of serine I put a choline out here on the right, I will have phosphatidyl-choline. If I put an ethanolamine out here, I will have phosphatidyl-ethanolamine. So anything that's phosphatidyl something means that I've got this part and then the something is attached to the phosphate. Okay? Pretty simple naming. Here's a variety of things showing that structure. There's phosphatidyl-serine. There's phosphatidyl-ethanolamine, and no, I'm not going to ask you to draw these structures, but I think you should know the general structure of a phosphatidyl compound. Okay? Here's phosphatidylglycerol. Here's phosphatidylinositol. Here's phosphatidylcholine. You saw a version of phosphatidylinositol last term when we talked about signaling. We talked about PIP2 and IP3. That was things that were derived from this. Was there a hand back in the back? Okay. Okay, so pretty simple structures. Another important class of molecules found in lipid bilayers are called the sphingolipids. That's S-P-H-I-N-G-O lipid, sphingolipid. This is a specific type of sphinglipid, and I'll say something about it in a second. Okay? Sphingolipids derive their name from the fact that they look like they're made from a compound called sphingosine. Sphingo and then S-I-N-E. Okay? There's sphingosine. And here's a sphingolipid. What are the differences between sphingosine and this sphingolipid? Okay? Well, there are a couple, but for our purposes, all we're going to be concerned about is the fact that this guy up here has an amine, and it has a hydroxyl. This guy down here has a fatty acid attached to that amine. Now, before I talk about this side of it, I want you to think about this side of it on the left. Oops, sorry. Okay? On this side on the left, what do we have? We have a long tail. There's 12 carbons, 13 carbons hanging off the end. Carbons and hydrogens, you know, are nonpolar. Therefore, this is have a nonpolar tail. Here's a fatty acid sticking off the end. That's going to be another nonpolar tail. And when we thought about the phosphatidyl compounds, they had two fatty acids hanging off the end. This looks like two fatty acids hanging off the end. Even though the structures are different than a glycerophospholipid, they physically resemble them. And that turns out to be important because there are structural considerations for what will fit into a lipid bilayer. Now, this is an unusual sphingolipid in that it actually has a phosphate. You see that phosphate there, you say, oh, that's just like the glycerophospholipids. Most sphingolipids do not have this phosphate. That's one way in which they differ from glycerophospholipids. Okay? This phosphate, you can see, has been attached to a molecule, and this molecule it's attached to is choline. So in this case, this red guy out here is choline. All right? Well, the overall structure is something we call sphingomyelin. Sphingomyelin is a sphingolipid. It's unusual in having a phosphate. Other sphingolipids don't have it. And it's a very important component of what's called the myelin sheath of nerve membranes. The myelin sheath. If you have trouble with your sphingomyelin, you're going to have significant problem with your neural membranes. Okay? There are many diseases associated with that. Sphingolipids tend to be found more in nerve and brain tissue than just in regular tissue, although you will find them to a limited extent in regular tissue, meaning skin and bone and so forth. But they're much more common inside of nerve tissue, as we find in brains. Well, the sphingolipids are unusual in that they can be quite complex. Here's a simple one, okay? So here's our one chain sticking off, here's our other chain sticking off, so there's our two nonpolar tails. Here we see no phosphate, and we see the attachment of a sugar. We don't see attachment of sugars to glycerophospholipids. We see them frequently to sphingolipids. Okay? Now, depending upon what is attached out here, we give these guys a new name. A sphingolipid that has only a single sugar attached is called a cerebroside. A cerebroside. Okay? Why do we call it a glycolipid? Well, glyco means sugar. Lipid meaning the nonpolar part. So this has a sugar and a nonpolar part. A cerebroside is a glycolipid. If instead of attaching a simple sugar to the end, we attach a complex set of several sugars, we create a molecule called a ganglioside. Ganglioside. So cerebroside has a single sugar, a ganglioside has a complex sugar arrangement. And I won't go through any here, but when we look at those later, you're going to see that gangliosides can be very complex. And gangliosides, like the ganglia in our brains, okay, or of our nerves, can be very, very full of these types of compounds.
What is the functional difference between the cerebroside and the ganglioside?
Yeah, good question, a common question. What's the functional difference between a ganglioside and a cerebroside? I can't tell you that. It probably relates to the structure. And that structure, as I can tell you, is very complicated in the case of a ganglioside, simple in the case of a cerebroside, and it's really not fully known why they are the way they are. But deficiencies in the metabolism of these can cause some very severe problems if they're genetic diseases in the cases of mental retardation and things like that. So very severe problems in the metabolism of these compounds. Okay, so I've given you two classes, and these are classes of molecules. Glycerophospholipids is a class, meaning there's many of them. Another class was the sphingolipids. You saw sphingomyelin, you saw cerebrosides, and you saw gangliosides. And there are many cerebrosides. There are many gangliosides. Okay? A third category of molecules that we find inside of lipid bilayers, cholesterol is a prime example. Okay? Everybody hears the word cholesterol, and the first thing they think of is bad molecule, causes problems. Okay? Well, our body has cholesterol because cholesterol is important for our membranes. Our body makes cholesterol because it's important for us. Cholesterol actually affects some of the properties of a lipid bilayer. It affects some of the properties of a lipid bilayer. It's not a trivial molecule. If we take brain tissue, for example, and we dry it out, and we weigh the components of brain tissue, what we discover is that cholesterol is 14% of the dry weight of a brain. That's a lot. 14% of the dry weight of a brain is cholesterol. Now we'll say more about cholesterol in just a bit. Looking at one more thing. Looking at cholesterol, it's mostly nonpolar. It only has one side that has any polarity, and that's that hydroxyl group right there. It's not a very polar molecule. Okay. I like this figure. Okay. This shows a hydrothermal vent in Yellowstone National Park. And these vents are kind of ugly. Okay. And inside of these vents where there's boiling water, there are microbes that live. Pretty hard to imagine, but they actually live in the environment of this boiling environment. Okay? These microbes that live here tend to have a slightly different kind of lipid in their membranes. Okay? Now, who can look at that and tell me what the structural difference is between that and the glycerophospholipids that I talked about before?
They're bonded to the glycerol by ether bonds, not ester bonds.
Okay, so there's 2 points of extra credit right there. Okay? That's the difference between that. There's ether bond right there. They're not ester bonds. And this ether bond here is stabler in the environment that these microbes are found. Okay? All right. This now schematically shows you what a phosphoglyceride, which is what I call a glycerophospholipid, or a sphingomyelin looks like, or in this case, an ether lipid. Archaeans are the names of the molecule, names of the cells that live in these vents. As you can see, at first glance, they don't look that different from each other. They resemble each other. And that's important because they all have to play together and work together inside of a membrane. Shorthand depiction. Polar, nonpolar. Okay. Well, if I take fatty acids and I mix them in water, I get something called a micelle. The micelle looks like this because they only have a single tail sticking off of them, a fatty acid and the polar end. Soap has this structure. Soap is basically fatty acids that have like a sodium put on them instead of a proton put onto them. Okay? Soap works because when you mix this with water, it forms this structure, a central part that's nonpolar, that is a place where grease can stick to. And it's carried away because the outer part of it is polar, and basically it's solubilizing grease in water. That's what detergents do. That's what soap does. These are called micelles. Okay? If you attach a second molecule to it, it can no longer arrange itself in that micelle structure. Instead, it spontaneously forms this lipid bilayer that I just described to you. So that second tail structurally changes what is stable. So lipid bilayers will form spontaneously if I mix them with water. There's a good structure there. And there's another good structure here. This is now showing some of these guys with unsaturated double bonds in them. Okay, unsaturated bonds inside of them give rise to things that look like this. Okay, well, liposomes turn out to be things that we can make in a laboratory very easily. We can make liposomes, and what is a liposome? A liposome is an artificial lipid bilayer that I can make by simply taking a bunch of glycerophospholipids and a bunch of sphingolipids, putting them in water and shaking them up. When I shake them up, they will spontaneously form this structure. And what's really cool about this is A, it looks like a cell, and B, on the inner part, that's water. Now, why is that important? Well, let's look at what I might do if I had a compound shown in red that's soluble in water and I wanted to get it into a cell. How would I do it? Well, one of the ways in which I could do it is I could take that molecule in red, mix it with a bunch of glycerophospholipids and sphingolipids, shake it up, and when the lipid bilayers form, some of them are going to contain my molecule. If I take these liposomes, and by the way, these are now called liposomes at this point. If I take these liposomes and I mix them with cells, they will actually fuse with them. Because the membrane structures are identical, they will fuse with the cell, and what will happen is these molecules that are inside the liposomes will end up inside of the cell. I've just created a way to deliver a molecule into a cell. So liposomes are very useful for this purpose. We use them mostly in the laboratory. It's not usually very easy to do in a medical environment because we have difficulty controlling which cells they go into. But in a laboratory environment, they work very nicely. Okay, if we try to measure the permeability, that is, the rate with which things move across the membrane, we're not going to worry about these numbers, but basically the further we go to the left, the less easy it is for this molecule to move across the membrane. We see that on the right side, which is the easiest, water is moving across fairly readily. Ions like sodium, potassium, chloride don't move across very readily at all. Glucose is somewhere in the middle, but even here, this is pretty difficult to move across the membrane. It's not fast enough, as I said, to maintain a cell and keep it alive by itself. Okay. Let's see, what do I want to say here? Okay. If I look at this figure, one of the other things that we discover inside of cell membranes are proteins. There are membrane proteins that are embedded to varying degrees in the lipid bilayer. We see A, B, C, D, E, and I'm going to give you some designations, some names for these guys. Something that goes across both layers of the lipid bilayer, like A and B do. Something that goes across both layers is called an integral membrane protein. An integral membrane protein is very difficult to remove from the membrane because it's stuck inside of there. Okay? If I have something that sticks with one layer of the lipid bilayer, but not both, it doesn't go through both, but in this case we can see it's stuck with this part of the lipid bilayer. Okay? This is what's called a peripheral membrane protein. So C is a peripheral membrane protein. If I have a protein that isn't even associated with the lipid bilayer, but it's associating in this case with another protein, I have a protein, an associated protein of the lipid bilayer. Okay? It's associated. It's not integral. It's not peripheral, but it's associated with it because if I look in the area of the lipid bilayer, I'll usually find this protein associated with it. Okay? The last category is shown by E. You see that E has a little tail sticking down into there. These are proteins that typically have a fatty acid attached to them, and that fatty acid really likes the inner part of that lipid bilayer. These guys are called anchored membrane proteins. We're going to see examples of all of these different kinds of membrane proteins. The ones that are involved in moving molecules from one side of the bilayer to the other are all integral membrane proteins, and we'll say more about them in just a bit. Okay. Now, I've got a little bit of time here, so let me just say a word about a really cool protein. And I'm going to bring this protein up for you right now, and later in the term, and probably next week, you're going to see why I think this protein is extraordinarily cool. Okay? It's called bacteriorhodopsin. Bacteriorhodopsin is an integral membrane protein. It projects through both layers of the lipid bilayer. And this molecule is involved in light sensing. So for bacteria that are photosynthetic, they will have this protein associated with them. And this protein does something really, really cool. In the very middle of this protein is a molecule of vitamin A. We know in our eyes that vitamin A is involved in vision. And so vision is a light-sensitive phenomenon. For bacteria, this is something that's allowing them to not only sense, but actually use light for a purpose. Okay? Well, how do they do it? Vitamin A is a molecule that has a long carbon-hydrogen chain, like a fatty acid. And it's full of double bonds. There are 4 double bonds in this long chain of a vitamin A molecule. And double bonds, you recall, can be in the trans configuration and in the cis configuration. And trans double bonds are physically different in structure than cis. Cis will tend to bend, trans will tend to be straight. Right? When you shine light on vitamin A, it causes trans and cis to interchange. So what happens is in the middle of this protein, there's this fatty acid-like thing that's sticking there. I'm going to use my leg as the example. Okay? My leg is presently in the trans configuration, but if I change one of the bonds in my leg at the place of my knee to a cis, it looks like this. Okay? Hope everybody can see my leg. All right? Trans, cis, trans, cis, trans, cis. When I shine light on vitamin A, it goes trans, cis, trans, cis, trans, cis, trans, cis. It goes very rapidly. And I'm using my leg for a purpose, because what this guy does is it kicks protons out of the bacterium. It literally kicks them out. Every time it bends, it kicks protons out. Now, we're going to see later that kicking the protons out turns out to be a really important thing for energy purposes. But for our purposes right now, all that's happening is that this vitamin A molecule that's inside of this bacteria rhodopsin, when you shine light on it, is kicking protons out. Okay? Remember that. I'm going to convince you next week that we can make a photosynthetic fish. Yes?
And then like the dark form, would it be all cis or a mixture of cis and trans?
In the dark form, what form is it going to be in? It'll be in whatever form it was left in when it became dark.
Are they all the same though, or are they different? Like, are some?
There are some variations between them, but for our purposes, we won't worry about that. In our eyes, there's what's called position 11 is the one that's involved in our vision, and it's that isomerization from cis to trans at position 11 that gives us vision. Yep. Okay. That's enough about those guys. I want to jump ahead and say something about prostaglandins. Okay? Another really interesting group of molecules that we talk about, and we'll say more about them later in the term, are called prostaglandins. And prostaglandins are molecules that arise from arachidonic acid. So that fatty acid that I showed you earlier that had 4 double bonds in it, it's called arachidonic acid. And arachidonic acid is converted into molecules called prostaglandins by action of an enzyme that you see on the screen. You see it is a peripheral membrane protein, meaning it's associated with one part of the lipid bilayer. And this enzyme is called prostaglandin synthase. Prostaglandin synthase. It's associated with the membrane, very importantly, because the place where we find arachidonic acid is only in the membrane. And it's found as part of the lipid bilayer. It's one of those fatty acids that's sticking off of these tails. Okay? Prostaglandins are really important for several things. One thing that prostaglandins are associated with is pain. Pain. When a bee stings you, and the instant pain that you feel, it's happening because arachidonic acid is being converted into prostaglandins because the bee causes the arachidonic acid to be released from the lipid bilayer, and this guy grabs it and makes prostaglandins out of it. Okay? This enzyme is an interesting enzyme because it can be inhibited by what we think of as non-steroidal Anti-inflammatory drugs. You probably won't call them that. You'll call them NSAIDs. N-S-A-I-D-S. NSAIDs inhibit prostaglandin synthase. They stop the enzyme from converting arachidonic acid into prostaglandins. They stop that process from happening. They are therefore known as painkillers. Aspirin is a painkiller. We have a headache, we take aspirin. And aspirin is stopping the synthesis of arachidonic acid in our brain or wherever. Painkillers are very good in this respect. Well, prostaglandins, if that's all they did, it would be very simple. But it turns out prostaglandins are involved in a variety of things. One of the things that they're involved in is uterine contraction. Another thing that they're involved in are inflammation. Yet another thing that they're involved in is the synthesis of tissue in our gut. They stimulate our intestines to make cells. When you hear about aspirin, people say, oh, I'm very sensitive to aspirin because it gives me stomach problems. What it's doing is it's inhibiting the synthesis of prostaglandins, and for some people, not all people, but for some people, this is a consideration because these prostaglandins, if they're knocked down enough, slow the synthesis of intestinal tissue. Intestinal tissue is some of the most rapidly proliferating tissue in your body. Because, why? Well, you got all those proteases in there that are digesting all those things. If you slow the synthesis of intestinal tissue, you're going to be much more likely to develop problems like ulcers and so forth. And for some people, that consumption of aspirin causes that problem. Now I want to tell you one more thing about these that are really interesting, and then we'll finish with a song. Okay? The other thing that's interesting about them is there are different kinds of prostaglandin synthase. People looked at this and said, hey, if we could just knock out the things that make the prostaglandins that cause pain, we'd be in good shape. Well, they discovered that there were different kinds of prostaglandin. And by the way, this prostaglandin synthase has another name. It's called COX, C-O-X. Okay? COX stands for cyclooxygenase, and we'll see later why that's called that. But COX enzymes are here. Well, there are COX enzymes that are called COX-1 and COX-2 and COX-3. And what they discovered was that there was a class called COX-2 that was found predominantly in joints. And that's where the pain was associated. And they thought, well, if we can make COX-2, that's the second type of prostaglandin synthase, if we can make inhibitors of COX-2 that don't affect COX-1, we're not going to have the side issues of intestines. And so they made these painkillers, and the drug industry spent billions of dollars developing these. One's called Vioxx, there's Celebrex and things like that. And what they discovered after they had made these and after they had gotten these patented was that some of them cause heart problems. Okay? Billions of dollars of investment all went bye-bye. Okay? All right. Well, that leads up to the song I want to sing for you today. It's to the tune of an old song called Oklahoma, and it's called Prostaglandins. Prostaglandins, the eicosanoids creating pain, are the ones to blame when you get inflamed and ouch, they hurt inside your brain. Prostaglandins, every throb and ache gets magnified. If you hope to win, cyclooxygens generations got to be denied. The Vioxx has all been recalled, so go get yourself Tylenol. And if you ache, plain PGH synthase, we must complain that you make the aches prostaglandins, prostaglandin, D2F1G2E2, prostaglandins, it's you. Okay, thank you. How's it going?
Good.
So do I need to give you my name for the extra credit?
Yes, send me an email. What would you?
Yeah, we'll do.
Tell me what it is here again. It's
John Myers.
John Myers, okay. Just send me an email, John. That'll remind me to put it in there, okay?
Oh, I got into pharmacy school, so thank you for giving that
Good.
shirt to the pre-pharmacy school.
Good for you. I'm happy to hear that. That's cool.
Hey, Dr. Ahern, did you hand this around? It looked like
not me.
Yeah,
Nope.
someone did, so I don't know.
Management and marketing internship.
Yeah.
Not me. Sorry. Yeah. How's it going, Naveed?
Hold on.
Hey, sir. Thank you.